Hi everyone, I'm Meghna. Today I'm going to present some recent work we've done on predicting homo-oligomer symmetry structures. This is joint work with a number of collaborators. I myself work at Microsoft, but I'm not rich. Franca was talking about rich people in the industry. Not everybody is rich. Compute is especially very difficult if you're at Microsoft because most of it is taken by OpenAI. Anyways, so I was not supposed to say that. Okay, so homoligomers are, we're going to talk about them. Most proteins function as oligomers. So these are complexes formed of multiple chains, and complexes with identical chains are homoligomers. Here are three examples of homoligomer proteins, and these are their symmetries. Cyclic 2 with two chains, so these are colored by chains. Cyclic with four chains, and icosahedral. Homologamization is essential in these proteins for biological function. So, for instance, with D-lactate dehydrogenase, which forms a homodimer, mutations that disrupt the dimer formation can result to instability and also, okay, careful. It can lead to instability and also loss of enzymatic activity. For a majority of proteins, the oligomer symmetry is not known, and traditionally solving these structures has involved X-ray diffraction methods, but these tend to be expensive. They cost about a few $1,000s per sample, and for a number of proteins, they are challenging because it's difficult to purify, crystallize, and stabilize the sample. And so structure prediction methods such as AlphaFold 2, Rosetta Fold 2, AlphaFold Multimer have been used for homoligomer structure prediction. However, the biggest challenge here is you have to try them out on different numbers of copies, exhaustively starting all the way from monomers to and dimers and trimers and so on. And then you pick the model which has the best scores in terms of PLDDT and other metrics and choose that as the structure. And so as you can see, this is a very computationally intensive process, and accuracy is typically only good on small homoligomers, so proteins that are shorter in length and that have fewer chains. And as soon as you go to longer proteins and proteins with more chains, you hit computational limits, and the accuracy also tends to be lower on proteins that have shallow MSAs. And so template-based methods, which are the cheaper computational alternative, have been also been popular. However, they require that the protein that is of interest to you should have a very strong homology to proteins with known homo-oligomer symmetries. And this is not true with a lot of a lot of understudied organisms, for instance. Other computational methods, such as quaternary state prediction methods, such as Queen, they predict only the multiplicity of the homoligomer assembly. So for instance, in this case here with the hemoglobin protein from Atlantic cord, the prediction from a quaternary state prediction method will be 4, but 4 is the number of chains in both a cyclic 4 homoligomer as well as a dihedral 2 D2 homoligomer. And so there is no way to disambiguate how exactly are these chains, how the layout is in 3D space. So given these challenges with the existing approaches, we propose a two-step approach. In the first step, we predict just the symmetry. So this reduces the search space for complex 3D structure methods such as AlphaFold. And then these predicted labels can be used as input to get the final 3D structure. Now, in the first step, the input to the approach can either be a protein sequence or an MSA. And then a protein foundation model is used. We consider ESM MSA, ESM2, and Rosetta Fold 2 models here. And beyond the protein foundation model block, there is a classifier head which is fine-tuned to predict oligomer symmetries such as C2, D2, etc. Now, where do we get this data, right? The labels C2, D2, and so on. There's no prizes for guessing. We get it from Protein Data Bank. So we scrape PDB and we get 130,000 unique protein structures with spanning 45 unique homo-oligomer symmetries. 8% of this data has multiple labels. So what that means is there are proteins that sometimes exist as dimers in C2 form, cyclic 2 form, and sometimes they might be in conformations that allow them to form higher order symmetries with 4 copies or 8 copies and in different layouts such as dihedral and so on. And given this data set of 130K unique proteins, we set up a training regime where we create splits using a sequence identity cutoff of 30%. What this means is that given a test protein, it's going to have at the most 30% sequence identity to any protein in the training or validation sets. In addition to this data set, we also set up 3 different held out sets for validation of our methods. So the validation split is used for hyperparameter tuning, for training, and so on. And these were completely unseen. So we didn't know what the results are going to be until we finally applied the selected models on these data sets. So this involves a test split from the PDB data set, a test set from the unifold paper, a set of homo-oligomers that was released by PDB in 2024. So these are novel structures, so they are challenging to predict on, and a set of designed proteins from the Baker lab, which is around 110 structures, which we call the de novo test set. Now, given the background and the data set, here are some problem formulation choices that led us to find the best model. We formulate protein homo-oligomer symmetry prediction as a multi-label classification problem with 17 homo-oligomer symmetry labels. We collapse rare categories such as C7, C8, C9, which are cyclic oligomers, into a single category, C7 to C9, for example. We consider all the protein foundation models in 2 different settings, either as pre-trained models where they are used as feature encoders, or as fine-tuned models where we train one or more layers, where we fine-tune one or more layers of the model. And since this is a setting with a very high class imbalance, we oversample the minority classes and undersample the majority classes. We also use cluster-based sampling during training to get heterogeneous batches. The loss functions we consider are binary cross entropy loss and margin loss. And area under precision recall curve is used because this is a class imbalance setting. So here are the results for this problem formulation and training regime. Our best performing model is a fine-tuned ESM2 based model, which takes only sequence as input, which we call SIG to SIM. And we notice that template-based methods perform the worst. Pre-trained models are better than template-based methods, and ESM2 based models are the best. We see a similar trend across the 2 other held out data sets that I spoke about. We find that SIG2SIM is more accurate than the popular template-based approaches like HH-search. HH-search uses hidden Markov models to compare a query protein against a database of proteins. And once we find hits between the templates, we assign the label of the protein from the database to the query protein. And here I'm showing the confusion matrices from both approaches. If you look at them, you'll find that the template-based method overpredicts majority classes. So since there is a higher prevalence of these, usually most of them get assigned monomer or dimer. And so it does very poorly on higher order classes. If you compare the model inference and training time, since SIG2SIM is only using protein sequence, it is computationally efficient compared to the MSA-based methods such as Rosetta Fold or ESM-MSA. And even when you consider end-to-end 3D structure generation, we find that it's faster than AlphaFold 2 based approaches. which have to do this exhaustive search where they search over all possible number of copies. And since we are avoiding that, we see substantial gains between 2 to 5 times, even from for end-to-end structure generation. So we can achieve proteome level predictions in hours as compared to weeks or months by prior work such as mol PC2 and this work by Shweke et al. We find that fine-tuned models are better than pre-trained models. And here, all bars that are above the x-axis are showing the performance benefit that you gain from fine-tuning the same model as compared to using a pre-trained version of it. And we see the biggest benefits for categories of symmetry that are very rare in our data set. Just to quickly give a sense of how class imbalance this setting is, the non-majority labels can be up to 100 times rarer than the majority ones. We find that sequence is a better representation in this approach of solving this problem than MSA. And we observe this by comparing the average performance of all of our sequence-based models to the average performance of all of our MSA-based models. The bars in gray show the performance of all the MSA-based models, and as you can see, they are lower than the sequence-based models. We do some qualitative analysis by analyzing the proteome level predictions on 5 different organisms, and we compare our results to prior work which has used AlphaFold 2 based prediction from this paper by Shweke et al. We find that they predict 44% of homodimers in E. coli. We predict a similar range, 48%. Their work predicts 21% homodimers in humans and yeast, and we predict the same. We predict 34%, which is higher than 21, but we predict the same number in both humans and in Homo sapiens and yeast. So we see some qualitative alignment as well to prior work. We show some additional results on a very large set of proteins from UniRef 50 in the paper. I will not go into these right now. Coming to the limitations of this approach, we find that Sig2Sim performs very poorly on de novo homo-oligomers. And as you can see here, the average AUC-PR is around 0.18 compared to 0.47 that we see on the PDV dataset. The template-based method also performs poorly, but it matches our approach in some of the classes. This leads us to ask, when does a model like Sig2Sim, which is based on sequence only and which is based on a protein language model, when do models like this perform well? And what we're trying to do here is characterizing the performance in the context of protein sequence homology. So what we want to do is understand the relationship between the model's performance and a test protein's homology to proteins that were seen during training. And as you all know, proteins are homologous when they come from a common ancestor. And typically, high sequence similarity over a long range of the protein implies homology. Here I'm showing a scale where homology is increasing from the left to right. And let's look at different points on this scale. Here is the point where the sequence identity is 30%, which means that a test protein is going to have at the most 30% sequence identity with any protein you have seen during training or validation. And this was our default training regime for this work, and which is where we observe this AUC-PR of 0.47 average across all symmetries. At the right extreme is a very easy setting where we look at cases where the sequence identity of the test protein is 95%, which means you will have certainly seen a highly homologous protein during training. And in this case, as you can imagine, it's an easy setting. So the model Sig2Sim gets an average AUC-PR of 0.643. Now, if you extrapolate between these performance numbers, you can kind of guess what the performance would be somewhere in between this range. So between 30%, which is at the edge of homology to the other extreme, which is certainly homologous. Now, to the left of this point here is what is called the twilight zone of homology, which means that there can be chance similarity between proteins that are not homologous. And this is sort of where the models like ESM2 and SIG2SIM start performing poorly. And at the left end is the setting where things are completely non-homologous, zero homology, and we see very poor performance here. All models perform very poorly here, and SIG2SIM also only does as well as the pre-trained models. So given this sort of analysis, this gives us a nice ballpark for what the future steps should be for this kind of work. We need to have approaches that do well on de novo proteins as well as native proteins. So things should work in the less homology regime. We need methods that are better streamlined in terms of end-to-end homology structure, homo-oligomer structure prediction. We find that oversampling the minority classes results in model calibration issues for us. And finally, some detailed analysis on the interfaces in the homo-oligomer structures made us realize that while the global symmetry looked all right, the interface residues were not always aligned well across interfaces. So to end, we make available the data sets, the code, and the models from this work. And I want to thank all my wonderful collaborators who I'm also working on other projects. Thank you.
Thank you very much, Meghana. We have time for one question.
Thank you. Interesting work. It happens that many proteins change their oligomerization state upon changes in the environmental conditions. So the information is not in the sequence, but is in the environment. How are you planning to approach this problem as the proteins might not have one oligomerization state, but several depending on the conditions?
Yeah, that's a great question. So unfortunately, protein data bank, we don't have that many such cases where for the same protein we see it in different control conditions. So I think data is the biggest bottleneck there, and maybe a combination of simulations, data from simulations, and some approaches like this might give you a reasonable answer to settings like that.
We have time for one more quick one. Very nice talk. I was wondering, your method, you said that the methods based on sequence alone are better than MSA ones,
Yeah.
which means the sequence is more conserved than the oligomeric state. But then your method performs worse for de novo sequences where you are lacking somehow the evolutionary background. So how do you explain this? What's your interpretation of this? Like, sequence is the best, but if either you go less evolutionary information is wrong, more info. How do you explain?
Yeah, that's a great question. So there are two reasons for this discrepancy. One is that the biological assemblies on PDB are not very accurate. So some of that, about 10% of them have a lot of noise. So we have some follow-up work where we remove those, but not everyone agrees on which ones are, ones where the lattice is not kosher. The other reason is that within an MSA itself, we see a lot of variance. So if you look at the homoligomer symmetry of all proteins within an MSA, you'll see they are like very, very different. And so that's the reason why MSA methods don't do well. As far as why ESM2 does not do well is, you kind of answer that question yourself. Okay.
Fantastic. Let's thank the speaker once again. Thank you very much.